Another application of these computational strategies is to study another biological process known as the programmed cell death, and this process is also known as apoptosis. So this process is very important in the living organisms, and during the development, certain cells should be dead while it is important in the developmental stages. So these special cells, they die when they are devoid of the special survival factors, and this process is also initiated by the expression of certain genes that are important in this process. So this process can be studied in the model organism C. elegans, and some other organisms, higher organisms, especially in nematode, and it is find that the some genes that are important in this process of apoptosis, they have identified certain genes, and if there is a mutation in these genes, especially in the brain part of these nematodes, then this process of apoptosis is reduced or completely eliminated. To find the genes that are involved in the process of apoptosis, and especially their products, means the proteins that are involved. So another strategy of mass spectrophotometry is used to identify the sequence of the proteins that are produced from those genes. So this process of mass spectrophotometer is very sensitive, and it has opened up a new experimental and computational possibilities for protein studies. So a protein can be digested into peptide by proteases like trypsin, as we have seen earlier. This strategy was used by Sanger in the sequencing of the insulin. So in a matter of seconds, protein can be digested into peptides. So for example, if we consider this protein and we use the enzyme trypsin, and this protein will be converted into small peptides. Then we use these small peptides and use another machine known as the tandem mass spectrophotometer, and this machine breaks the peptides into even smaller peptides in a very short time, and it measures the mass of each of these small fragments. The mass spectrum of the peptide that is obtained is the collection of the masses of these fragments. The protein sequencing problem is to derive the sequence of the peptide given in its mass spectrum. For an ideal fragmentation process where every fragment of the peptide is generated, and in an ideal mass spectrophotometer, the peptide sequencing problem is simple. However, the fragmentation process is not ideal, and mass spectrophotometers measure mass with some imprecisions. These details make peptide sequencing difficult. A mass spectrophotometer works like a charge sieve, this instrument. A large number gets broken into smaller fragments that have an electric charge. We know that proteins have an electric charge. These fragments are then spun around and accelerated in a magnetic field until they hit a detector. Because large fragments are harder to spin than small, one can distinguish between the fragments with different masses based on the amount of energy required to fling the different fragment around. So it happens the most molecules can be broken into several places, generating several different ion types. So these ion types, the problem is to construct the amino acid sequences of the peptide from the masses of these broken pieces. So this strategy is used by using the tandem mass spectrophotometer, and it measures the amount of the ion present in the smaller fragments, and these can be used to sequence the proteins of different sizes.